morning, everyone. Welcome to the Department of Medicine Grand Rounds on this chilly Thursday morning. I'll be standing in for Dr. Williams this morning to introduce our guest speaker today. Today, we have none other than Dr. Alex Carll. Dr. Carll, by way of a brief background, spent a lot of his time on Tobacco Road. Don't hold that against him. I'll promise not to say the names of the universities, but he spent time as an undergraduate and a graduate student at the one in Durham and the one in Chapel Hill. He has degrees in environmental science, toxicology, public health, and as you will see from his talk today, all of those come to bear on his program of investigative interest. After his formal training through his Ph.D. years, he then moved to Boston, where he studied at the Chan School at Harvard as a postdoctoral fellow. Then, after a couple of years of his fellowship, we were fortunate enough to recruit him to the University of Louisville, where he came about 10 years ago. Since that time, he has established both a regionally and nationally recognized program related to cardiovascular toxicology. In particular, he's been recognized by the National Institutes of Health by multiple rounds of NIH-supported funding that's very much related to the topic of discussion today. He's also become a regular fixture in the Society of Toxicology, and as you might guess, particularly in the cardiovascular toxicology subsection. He, during his time at the University of Louisville, has turned out already a couple of PhD students. One of them is a physician at the Mayo Clinic. Another moved out to the West. He has another one probably to graduate maybe in this calendar year, and certainly has a lot of interest for more students to join his lab soon. In addition to all of this, Dr. Carll also serves on various society leadership boards in terms of being a counselor in the Society of Toxicology and other relevant bodies. He has also quite recently, just several weeks ago now, been formally moved to the position of associate professor with tenure. Kudos, Dr. Carll. Congratulations on your well-deserved promotion. And with that, I'll turn it over to you so we can learn from you about your program and autonomic regulation and e-cigarette cardiovascular toxicity. Dr. Carll.
Well, thank you, Steve. And thank you all for joining today, braving this extreme cold, the blizzard and the flooding to meet virtually. I'm going to talk about my research focused on e-cigarettes and how ingredients within e-cigarettes, as well as how the autonomic nervous system might possibly mediate their toxicity. But before I continue, I gotta figure out how to, there we go. Before I continue, I just want to state that I have no conflicts to disclose. A few disclaimers. The closest brush I've had with a conflict was when Juul Labs, which manufactures one of the most popular e-cigarettes around, solicited me in 2018 and offered me a little bonus to continue my work at University of Louisville with a $200,000 a year payment. And needless to say, I declined, but I got some free publicity out of that. And then also want to note that this research was supported by the NHLBI and FDA, but the content is solely the responsibility of myself and colleagues and doesn't represent the official views of NIH or FDA. So a little background on electronic cigarettes. In case you missed it, over the past 15 years, they've become exceedingly popular, and that's especially true among youth. And so really the peak use was, for among youth at least, achieved right around in 2019 with about a quarter of high schoolers reporting vaping within the past month. And this rise in the use prevalence is largely attributable to Juul, which designed a very sleek device that was relatively discreet, easy to hide, and also kind of sexy. The company did an excellent job of advertising, especially to youth, for which they were successfully sued. And then clearly there was a successful uptake in use. So another thing not mentioned about Juul is that they designed a new nicotine formulation that was especially palatable, and as a consequence, their device became exceptionally addictive. So since 2020, use prevalence has continued to drop among high schoolers. We're about at a half the prevalence that we had in 2019. But middle school use prevalence has started to rise again. And not shown in that graph is that actually among the young adult population at 18 to 34 years of age, there has been a rise in use prevalence. Some have asserted that this is displacing cigarette smoking, and that's sort of what's depicted here in this graph. But a little caveat about this graph: the projections, all the data were generated in a publication that was funded by Juul, and the manuscript was revised by Juul. The authors were consultants for Juul, and so you had to take it the smoking data with the e-cigarette data with a grain of salt. But the thing I want to focus on is that even though the use prevalence among youth may have dropped, basically the users of 2018, 2019 are aging into adulthood, and it's likely that use prevalence of e-cigarettes among young adults is only going to continue to rise as well. So some major questions for e-cigarettes are, firstly, are e-cigarettes actually supplanting smoking? Well, we're not going to address that today, but it's certainly a relevant question to their public health harms or benefits. But another important question is, are they a safer alternative to smoking? And what are their cardiovascular and cardiac impacts anyways? Before I can get into that, just a little bit of a further introduction on e-cigarettes. There are a number of designs that have sort of evolved over time. The leftmost being the first generation, all the way to the rightmost being the Juul fourth generation. And not shown here is a fifth generation, which is very similar to these pod devices, but they're called pod mods. But suffice it to say that all of these devices operate with a battery. Some of them allow temperature or voltage control. Some of them produce much thicker puffs, like the mod device. And some of them allow continuous temperature control to avoid any potential burning of the e-liquid itself. But they all contain e-liquids. And within those e-liquids, more times than not, they contain nicotine, flavors, and solvent vehicles. So the solvent vehicles themselves generally are composed of a mixture of vegetable glycerin, or VG, and propylene glycol, or PG. And usually those are at about a 50-50 ratio to each other, or 30% VG or 70% VG, with the remainder being PG. But when you heat these solvents, they generate aldehydes, particulate droplets, and reactive oxygen species. For VG, it disproportionately produces acrolein and formaldehyde. And for propylene glycol, it disproportionately produces acetaldehyde. Why is that important? Well, a somewhat recent study estimated that about 92% of smoking's cardiovascular risks are attributable to acrolein, acetaldehyde, and formaldehyde. And as mentioned, these are produced by the solvents in e-cigarettes, and they're also found, not surprisingly, in the aerosols of e-cigarettes, but usually at lower levels than observed in cigarette smoke. In addition, studies not involving e-cigarette particles emitted in their aerosols, but other types of particulate matter have shown without a doubt, really, that inhalation of fine particulate matter causes autonomic imbalance and arrhythmia. And acrolein itself has experimentally been demonstrated to cause imbalance of the autonomic nervous system and induce bradyarrhythmias, or sort of skipped beats that slow the rhythm temporarily of the heart. Meanwhile, nicotine, if you weren't already aware, is a well-established sympathomimetic that has been shown in a multitude of studies to evoke arrhythmias, and also it's indicated to increase risk for sudden cardiac death. In terms of sudden cardiac death and electrophysiology, would imagine as clinicians, most of you are well familiarized with the electrocardiogram. You probably call it an EKG. Usually,
Please.
yes. Okay. Usually in experimental toxicology, we refer to it as an ECG. So bear with me because I'll call it ECG throughout the rest of this talk. But if you're not aware, the electrocardiogram is an exceptionally informative tool about the potential risk or underlying disease of an individual. And we can assess changes in the shape of the electrocardiogram and sort of the cardiac conduction cycle to indicate that. But we can also look at heart rate variability to assess changes in regulation by the autonomic nervous system of the heart. And in addition, we can look for disruptions in the regularity of rhythm of the heart, termed arrhythmias. So shown here is just sort of a cartoon of a human heart that is color-coded to the electrocardiogram to remind you that the P wave on the electrocardiogram is when the atria depolarize. The QRS is when the ventricles depolarize, and the sort of ST interval is when the ventricles are repolarizing. And we evaluate the electrocardiogram for a number of different intervals and amplitudes, but one of the most commonly used indexes of the ECG is the QT interval. And that's because prolongation or shortening of the QT interval can be especially predictive of cardiac arrhythmias, so much so that the FDA mandates testing of QT interval for all experimental drugs being developed. And also QT prolongation is one of the leading causes of withdrawal of drugs from the market and cancellation of clinical trials. So in mice, the electrocardiogram looks a little bit different, but the similarities are probably greater in my opinion. They have a P wave, a Q, an R, an S, and a T. And they also have a J wave that is pretty consistent and usually prominent. Actually, a lot of times it's more positive to the extent that it's often confused by researchers as a T and published and reported as such. But the T wave itself in the mouse is actually inverted and it's very subtle. So it's very challenging to measure accurately to get a QT interval, for instance. So we use the electrocardiogram also to quantify arrhythmias. And we quantify a number of different types of arrhythmias, but one of the most consistent ones that we observe is referred to as a premature ventricular contraction. In this talk, I will refer to it as ventricular premature beat, but they are synonymous. And they're important because they are associated with cardiac mortality. So generally, PVCs are in the clinic perceived as nothing of great concern, but certainly, you know, they're pretty common. I think all of us probably have them at this age to some extent or another. However, a somewhat recent epidemiologic study showed that in patients referred for Holter monitoring, individuals who had 12 or more PVCs or VPBs per day had a clearly higher risk of cardiac mortality over the ensuing 10 years after their measurement. And the difference was especially stark for sudden cardiac death, which you can see is 6-fold higher risk for those with greater than 12 ventricular premature beats per day, which is really a half of one of these arrhythmias every hour. So we also analyze the electrocardiogram for heart rate variability, which is an index of autonomic imbalance or autonomic balance itself. So shown here on the x-axis, I've put heart rate variability, and on the y, cardiovascular risk. And that's because a plethora of studies have shown that heart rate variability correlates inversely with cardiovascular risk, such that a low heart rate variability is associated with markedly increased likelihood of death or disease for cardiovascular complications. But also importantly, low heart rate variability is consistent with sympathetic activation. So sort of the fight or flight stress responses or an enhanced basal state of adrenergic regulation. And conversely, elevated heart rate variability is consistent with parasympathetic dominance or the rest and digest control of the body. And this is generally perceived to be a healthy thing, although extremes can lead to arrhythmias themselves and can also occur in certain patient populations. So how do we derive heart rate variability? Well, shown here is a human lead-2 electrocardiogram with successive QRS complexes on an ECG, and you can see that there are a multitude of RR intervals. We measure those intervals, and they have very subtle differences in length that are depicted here on this scattergram across time. So this is a 5-minute ECG waveform, and the RR intervals are distributed at different lengths. The mean is indicated by red, and the blue indicates the standard deviation around the mean in that 5-minute period. And that generates the SDNN, which stands for the standard deviation of normal to normal R to R intervals. And meanwhile, we also get the RMSSD, which is the root mean squared of successive differences. These two both go down with sympathetic dominance, and they go up with parasympathetic dominance. So again, a higher heart rate variability is usually an indication of better cardiovascular health. In addition, we generate the frequency domain parameters through a fast Fourier transformation, and high frequency heart rate variability is pretty much analogous to RMSSD, although not superfluous. So we measure that as well, such that if it goes up, it's parasympathetic dominance, and down is sympathetic dominance. LF is a little bit trickier, and I don't rely on it because of a number of different other factors that influence it that seem to be independent of the autonomic nervous system. So with respect to smoking and e-cigarettes, the pre-existing data indicate that, firstly, cigarette smoke, not surprisingly, increases sympathetic modulation after exposure. And this has been shown in humans, but shown here is in mice from a group at UC Davis, where they exposed the mice for 4 days and monitored them immediately after exposures. And you can see the depressions in RMSSD as well as SDNN. And more recently, Holly Middlecoff's group looked at the impacts of e-cigarette use in humans shortly after they puffed on an e-cigarette. And they found that the HF parameter was depressed, but only if there was nicotine in the e-cigarette versus e-cigarette without nicotine and sham control, which I believe was a straw. So there is pre-existing data indicating that e-cigarettes and smoking cause sympathetic dominance. And a cardiology fellow who got his Ph.D. in my lab took a Louisville Healthy Heart Study cohort and analyzed those individuals, including 53 smokers, for their electrocardiograms to see if perhaps the autonomic nervous system was related to any changes in the ECG. So these individuals had their cotinine measured from urine, cotinine being the primary metabolite of nicotine. And he observed, first and foremost, that cotinine inversely correlated with the PR segment. So that's the atrioventricular depolarization time. But he also importantly found that dopamine in the urine was inversely associated with the PR segment. And actually, through statistical mediation analyses, dopamine mediated the relationship between cotinine and PR interval. So dopamine is acetylcholine, and it's very much related to sympathetic activation. It's the precursor to norepinephrine. And so this data indicates that the autonomic imbalance or sympathetic dominance promoted by smoking has impacts on the electrocardiogram as well. We followed this study up by looking at the NHANES 3 population, which had 5,633 individuals, smokers, and we looked at the cotinine levels in their serum and also saw PR segment shortening with increasing cotinine levels. So that's not the only study or set of studies that have shown ECG effects of tobacco use. More recently, Holly Middlecoff's group showed not only that tobacco cigarette smoking acutely prolonged the T peak to T end interval when normalized by QT. So this is an index of heterogeneity of ventricular repolarization and therefore sort of risk for arrhythmia. Also, nicotine-containing e-cigarettes significantly elevated this index, but e-cigarettes that did not have nicotine had no such effect. So what about the longer-term impacts? In a more recent study, Irfan, and I looked at NHANES 3, not only for the relationship between cotinine levels and sort of linear changes in the ECG. We also looked at abnormalities in the ECG and found that among smokers, cotinine was associated with an increased risk in long JT interval and short QRS duration. And then with a median 22-year follow-up in that population, we found that this short QRS duration in smokers and the long JT interval were both predictive of an increased risk of cardiac or cardiovascular mortality. So the changes in the ECG that we observed in conjunction with cotinine and nicotine and therefore smoking can be predictive of cardiovascular mortality. And this is just a series of survival curves for non-smokers on the left and for smokers on the right, without all of the covariate adjustments. But just to kind of clarify, this is the long JT population among smokers with clearly depressed survival compared to the other groups. Okay, so back to the mice research that I do. The overarching hypothesis of my work is that e-cigarette aerosols change the physiology of the heart through imbalance of the autonomic nervous system and individual chemical constituents within the e-cigarette itself. And to get at that, we use radiotelemetry more often than not in these mice. So we surgically implant them with radio transmitters that allow us to continuously monitor ECG while the animals are conscious and fully recovered from surgery and exposed to these aerosols, but also while they're in their home cages. And shown here is just a magnified ECG from a mouse, and just kind of clarifying that we can get virtually all of the intervals from a mouse, including the QT interval, but also PR interval and PR segment and QRS, through analysis that we perform with the specialized software IMCA's ECG Auto. So first, I want to get into the acute effects of e-cigarette aerosols on rhythm and rhythmicity. But I just want to hopefully, let's see, show you the setup for exposures. All of our exposures involve 9-minute puff sessions, alternating with 9-minute washouts, with 4-second puffs every 30 seconds. So there's 18 puffs within a 9-minute span. And this is to simulate human vaping. So shown here is a Joule device connected to the in-exposed Cyrex system. And you'll see the aerosol goes into this mixing chamber. These are the air-exposed animals while we're monitoring them with telemetry. But over to the right, you'll see in a second the e-cig exposed animals. So the aerosol is generated. It is delivered by a pump system over to the e-cig exposed animals who are being monitored real time with telemetry. And so the aerosol dissipates rapidly after the puff, usually within about 10 seconds. And so we usually have about a 20-second period that's relatively aerosol free before the next puff in a 9-minute puff session. So initially, we saw that, let's see, acute exposures to e-cigarettes depended, sort of their effects depended on the presence of nicotine. And we found that when we delivered aerosols just from the solvents themselves, aerosolized in a first generation device with sort of a second generation like cartridge that we could refill, when we delivered just the solvents, it induced significant bradycardia in these mice. Conversely, when we use the commercial cartridges with either tobacco, classic tobacco, or magnificent menthol from Blue, the effects were not nearly as stark, although you can see a similar bradycardia in these initial exposures to some extent. And then importantly, after exposure, all exposed mice had elevations in heart rate relative to the air control, which you can see here, which had kind of a gradual decline over the course of the exposure because of acclimation to the exposure chamber at that moment. We compared males to females, and we found that the males had much more dramatic responses than the females. And so within the males, this is just sort of a waterfall plot that depicts 1 minute averaged ECG waveforms for every line. So these are successive minutes going from the back forward. And so in blue is before exposure to PGVG aerosol, and in black is after exposure to PGVG aerosol once heart rate had returned to relative normal. And you can hopefully appreciate that we had QT prolongation, JT prolongation, indicating slowed repolarization, PR prolongation, a number of effects with this exposure. But interestingly, we did not see a significant elevation in arrhythmias from the PGVG alone when it came to ventricular premature beats. However, we did see significant increases in ventricular premature beats with this magnificent menthol aerosol and with the 100% PG aerosol. Typically, PG aerosol is not, or PG at 100% is not really a thing that's used, and that's because the efficiency of aerosolization is pretty low for PG, so you need to cut it with vegetable glycerin. But also of note, shown here, we compared to mainstream cigarette smoke, an ultralight like research cigarette, and a sort of full flavor Marlboro Red like research cigarette. And we didn't see ventricular premature beats with those exposures, nor did we see increased VPBs with exposure to acrolein itself. So that was a first generation like e-cigarette. And we wanted to study the impacts of kind of the e-cigarette that was really taking off as of 2018. And that's what this graph shows here, is just use prevalence among high school students. And one thing that really sets the Juul device and sort of everything that's come since apart from these past e-cigarettes is the higher nicotine content. And the manufacturers of Juul were able to achieve this by protonating the nicotine in a salt formulation, which made it less aversive to inhale at higher levels, and therefore much more tolerable to users to get a bigger hit per puff of nicotine. Well, right around when Juul was reaching its peak use prevalence, Michelle Gonowitz showed that among adolescents who were solely using e-cigarettes, they had 58% higher urinine cotinine than had been observed in adolescent smokers. And so this suggests that these kind of more efficient e-cigarettes may be more addictive and in some respects more risky than conventional smoking itself, at least with respect to nicotine. In addition, there were a number of different flavors that were available with Juul and remain available with other products, and these might bear unique toxicity. So we wanted to look at the acute effects of Juul aerosols, and to do so, we performed a series of 15 puff sessions indicated in red, 9-minute puff sessions that were punctuated by 9-minute washout periods. And we exposed telemetered mice. This is a small scale study, but we exposed them to air as a control, or PGVG with or without nicotine. And that nicotine was nicotine benzoate at the same level as other exposures involving Juul Virginia tobacco or Juul menthol. And we found that, similar to our observations with magnificent menthol from Blue, Juul menthol also significantly increased ventricular premature beats, whereas the other aerosols did not achieve this effect. In addition, when we analyzed heart rate and heart rate variability according to either the washout period or the mid-exposure period, we saw a clear elevation in heart rate during the washouts. And it's important to distinguish between that mid-exposure period and the washouts, because as you might recall, we had dramatic bradycardia during those 9-minute puff sessions, but afterwards a rise in heart rate. So Juul menthol was especially effective at increasing heart rate, but also it significantly depressed heart rate variability during these washout phases. In addition, we looked at the lungs for the ratio of GSH to GSSG. So this is an indication of potential oxidative stress or depletion of antioxidants. And we found that Juul menthol uniquely depressed this ratio, suggesting oxidative stress in the lungs. So we evaluated the contents of the e-cigarette aerosols produced from these different arrangements, and we found that just bubbled extract had 72% and 40% higher acrolein and formaldehyde, respectively, if from the Juul menthol, relative to the Virginia tobacco e-cigarette. But also we looked at plasma aldehydes, and we could see that menthol produced or induced a significantly higher level of plasma aldehydes in mice that were exposed for 6 hours to these aerosols. So it appeared that Juul menthol was disproportionately producing formaldehyde and resulting in increased formaldehyde and aldehyde uptake. And that's important because we had some pre-existing data looking at the impacts of formaldehyde itself. We, in this study, exposed mice for 14 days successively to formaldehyde at 5 parts per million, 6 hours per day, and analyzed both heart rate variability on select days, but also the occurrence of ventricular premature beats. And we saw that towards the end of the exposure regimen, there were significant elevations in these arrhythmias. So formaldehyde in and of itself is arrhythmogenic, and it's important to distinguish these observations from those acute effects that I just shared with you. Clearly, there's no nicotine in this study, and there aren't a bunch of particles and other aldehydes. There are a lot of constituents in the e-cigarette aerosol that just aren't here in this exposure. But the point is that formaldehyde itself is arrhythmogenic, and it might underlie some of the effects of e-cigarettes. We looked further at the impacts of formaldehyde by examining these mice after the culmination of their 14-day exposure regimen. We looked at their responses to a restraint stress. So we placed the mice inside of this restraining tube for 5 minutes and looked at their heart rate responses to restraint, finding that the formaldehyde exposed group actually had a more rapid decline in heart rate after they were released from the restraint tube, indicating sort of an enhanced parasympathetic rebound in this group. Not something we really expected, but we looked further at the heart rate variability, and this seemed to affirm an enhanced parasympathetic rebound in these formaldehyde-exposed mice. So just remember that as we move forward, I'll show you some other data with e-cigarettes that's going to tie back into this. Also, we looked at ventricular repolarization using a specific correction formula for each mouse and observed that there was a relative QT prolongation in the formaldehyde-exposed animals after this 14-day exposure. So it suggests formaldehyde itself is pro-arrhythmic and arrhythmogenic and alters autonomic regulation. Well, what about nicotine itself and also the different device types? We did perform a small-scale study comparing the Juul device to this box mod device that allows for really thick puffs of aerosol at a higher wattage. And we normalized by the nicotine concentration and saw that the box mod device at 2.7% nicotine and 5% nicotine induced comparable incidence rates of ventricular premature beats. And one little disclaimer about all these observations: the frequency of these ventricular premature beats is not gobsmacking. It's a relatively subtle effect, but across a massive population of e-cigarette users, these may actually translate to clinically relevant outcomes, especially in individuals with underlying susceptibility. The point of this data, though, is that there appears to be a threshold effect of the nicotine after which you kind of saturate its impact on arrhythmogenicity. The involvement of VOCs in these effects is unclear, but suffice it to say, nicotine is an arrhythmogenic constituent in e-cigarette aerosols. So what about nicotine's arrhythmogenicity outside of aerosols? This is just like a little bit of a translational segue. I got an email out of the blue from a paramedic who said he noticed he was having palpitations, and he checked his blood pressure. It was a little bit elevated, but relatively normal. And then he borrowed his coworker's Apple Watch and discovered that he was throwing PVCs repeatedly. He reported that he had been a regular tobacco user. He didn't specify whether he was a smoker or if he used chewing tobacco, but he had switched a year ago to nicotine pouches. So Zyn is one of the most popular nicotine pouches out there, and just want to note that Zyn itself contains nicotine benzoate, this kind of formulation that allows for much higher tolerable nicotine levels. And it may have been the culprit. We don't know. That's just sort of one example. But my PhD student, Corey Kucera, set out to test the different nicotine types in mice. He compared the conventional free-base nicotine, which is more common in the traditional e-cigarettes, to nicotine salts and synthetic nicotine itself using a racemic mixture. So there are different isomers of nicotine. The S isomer is the natural occurring one in tobacco, and that's present in both freebase and nicotine salt devices. But then more recently, synthetic nicotine was developed to skirt FDA regulations that have since been revised. And that has a combination of both S and R isomers. So we set out to test whether the nicotine types differ in toxicity. We exposed telemetered mice in a crossover fashion to these different aerosols of different nicotine types across a series of different days. So each exposure to a given nicotine type occurred with a 3 to 4 day interval between the other nicotine type exposure. And the concentrations were increased sequentially after 3 puff sessions each. We found that nicotine salts or nicotine benzoate, what's in Juul and most of these modern e-cigarettes, was especially effective at inducing increases in heart rate relative to the air control and the vehicle control. It also uniquely induced reductions in heart rate variability. So both of these collectively suggest a sympathetic dominance. In addition, nicotine salts increase ventricular premature beats and were the only sort of formulation to significantly do so. We did a follow-up study where we pre-treated mice with either normal drinking water or with drinking water containing atenolol, and we found that the impacts of the 2.5% nicotine benzoate, which previously was not arrhythmogenic, were abolished. So there was no increase in heart rate or reduction in heart rate variability when atenolol was provided to these mice, which confirms that this was indeed a consequence of sympathetic activation of the beta-1 adrenergic receptor. We also found that nicotine salts induced a greater intake in nicotine itself. So shown here, 5% nicotine salt when compared to 5% freebase in animals exposed to the same aerosols, just different nicotine, clearly resulted in an increase in total nicotine equivalents in the blood of these mice. So it's more effective at reaching the lungs for one reason or another. It may be due to behavioral responses of the mice, aversion from the 5% concentrations in the freebase nicotine. It's not entirely clear yet to us. Okay, so back to flavored e-cigarette aerosols and Juul menthol. I mentioned this earlier. We had found that Juul menthol induces a sympathetic dominance acutely using heart rate variability. But we also followed up by assessing catecholamines in urine and found that epinephrine was elevated as a consequence of Juul menthol exposure. We also evaluated the acute effects in mice with both blood pressure and ECG telemetry concurrently, and saw that ventricular premature beats were elevated with a single acute exposure to Juul menthol. And then in a separate pilot study using a different kind of mice, 129 SVs, we pre-treated these mice with propranolol, a beta-1 and beta-2 inhibitor, and found that relative to just typical drinking water, the propranolol abolished the arrhythmogenic effects of Juul menthol. We then followed up in C57s, which is what we have been reporting all of our data on in the previous slides, and saw that beta-1 selective inhibition with atenolol also prevented these arrhythmias, indicating that the arrhythmogenic effects of e-cigarettes was mediated by beta-1 adrenergic activation. Okay, so what about the impacts of repeated exposures? We've looked a lot at acute exposures, but we wanted to know what might happen in mice with a series of repeated exposures. So we performed a 20-day inhalation exposure in telemetered mice with ECG and blood pressure telemetry, and had a concurrent air control group that was exposed to just air on the same days for the same durations. And each of these exposure days involved 10 puff sessions. We found that when analyzing selectively for the first and the last day of each week, just because the workload for the full analysis was quite extensive, that heart rate was only significantly elevated on day one, although you can kind of see an apparent trend of elevated heart rate on the subsequent days. These weren't significant. And then conversely, blood pressure was pretty clearly elevated on all of the days, with significant effects on sort of the initial 10 days, and then trends of significant elevations on the subsequent days. And this may just be due to our limited number of successfully telemetered animals. But one thing to kind of bring into mind here is that a recent study by Holly Middlecoff's group looked at e-cig users with one acute use of an e-cigarette and found that there were similar impacts on blood pressure. I'm sorry, that's heart rate there. Similar impacts on blood pressure that were significant in their participants. Mind you, these measurements were taken after exposure, where we're seeing the impacts being a bit more pronounced in the mice during the exposure. But also importantly, these blood pressure effects were not really temporally associated with the impacts on heart rate or arrhythmia. In terms of arrhythmias, the arrhythmogenicity was relatively transient and dissipating with successive exposures. So the initial exposures were particularly arrhythmogenic in these mice. Well, we wanted to look to see if there were any adaptations in the heart in terms of protein expression or protein phosphorylation. So we looked at hearts using phosphoproteomics at both 5 days after exposure and 20 days after exposure. And in terms of the 5-day post-exposure mice, we observed a number of pathways that were enriched using ingenuity pathway analysis, including signaling related to dilated cardiomyopathy, beta adrenergic signaling, dopamine, and pKa signaling, all sort of pointing the finger at sympathetic activation from these exposures. But there were another series of changes that weren't necessarily attributable to adrenergic activation. We also performed a toxicity pathway analysis and found that proteins were enriched related to cardiac dysfunction, enlargement, dilation, and arrhythmia, as well as heart failure. And then at 20 days, the story was similar. There wasn't as sort of blatant of an indication of beta adrenergic activation, but we did have enrichment in pKa signaling. And you can see insulin receptor signaling, a number of certainly interesting observations here. What we're going to do with this, we're not 100% certain, but we do have some ideas about how we're going to pursue a particularly arrhythmia-related protein, PITX2, in subsequent studies. But anyways, the toxicity pathway analysis in this 20-day exposed group showed a similar story as well. In terms of their physiology towards the end of the 20-day exposure, we did see depression in heart rate before the exposures on any given day in that final week. And this was recapitulated 1 day after cessation of the exposure in the Juul menthol exposed mice, where they had depressions in heart rate. Surprisingly, even after 23 days following the cessation of exposure, the Juul menthol exposed mice had a depression in heart rate of about 130 beats per minute. And when we stressed them with restraint, we saw, again, this kind of more rapid rebound in heart rate that was semblant to the impacts of formaldehyde itself. In addition, we observed a QT prolongation that could be arrhythmogenic. We didn't see increases in arrhythmias at this 3-week time point following the cessation of exposure, but it indicates potential ventricular remodeling. Note that at the time point of our proteomic analyses and heart collection, we did not see a QT prolongation. So we might have missed an opportunity here, but we didn't have a 3-week post-exposure set of hearts that we could analyze for this purpose. So in summary, our data indicate that there are numerous potential mediators of cardiac arrhythmias and dysfunction, and these include menthol, as well as the device type, nicotine type and levels, and beta adrenergic receptors, as well as formaldehyde. But to sort of follow up on the flavorants themselves, one constituent in e-liquids that's not really regulated as a characterizing flavor is called a coolant. It's really a constituent class. And these coolants have become exceptionally popular in e-cigarettes, especially the ice-flavored e-cigarettes. They're found almost universally in e-cigarettes at often very high levels for any of the other constituents. They're the predominant one when it comes to flavors or additives. And so my PhD student, Corey Kucera, set out to test whether coolants exacerbate the autonomic effects and the arrhythmogenic effects. And again, we use this kind of crossover design with a series of exposures to different coolants or vehicle control, this time at 2.5% nicotine benzoate. And we followed a similar design to that used for the nicotine exposure study, where we increased the concentration of that day's coolant gradually across the course of that exposure. We found that the coolants, especially WS23, increased the impacts on heart rate of e-cigarettes, and as well, the impacts on heart rate variability. And this was especially evident at the washout phases and consistent with sympathetic activation. But also we found that WS3 and WS23 e-liquids both significantly elevated ventricular premature beats relative to air and also vehicle. And when we looked at the ventricular premature beat occurrence during the different concentrations, the impacts were especially clear for WS23 at the highest concentration. We also analyzed the heart rate changes for any potential correlation with the occurrence of these arrhythmias, and we found that especially for WS23, but in fact for all of the e-cigarette aerosols, an elevation in heart rate was correlated with the increase in ventricular premature beats, such that a 50 beat per minute increase in heart rate corresponded for WS23 with about a 350% increase in ventricular premature beat incidence rate. We also evaluated the QT interval and didn't really see any clear average effect across the groups. But importantly, we did see that the occurrence of these arrhythmias, they're relatively infrequent, but within an individual animal, the occurrence of these arrhythmias, if they were exposed to a WS23 aerosol, was significantly correlated with QT prolongation, suggesting collectively that sympathetic activation and changes in ventricular repolarization may underlie the arrhythmogenicity. So we wanted to kind of tease this apart a little bit further, and we performed testing in human-induced pluripotent stem cell-derived cardiomyocytes, collaborating with Matt Nestoriak. We first sort of ground truth that norepinephrine itself would reliably increase beat rate and shorten the field potential duration, which is analogous to QT interval in these cardiomyocyte-like cells. And we saw, sure enough, that norepinephrine shortened field potential duration and increased beat rate. And it did so in a relatively consistent relationship, for which we were able to develop a new correction formula, somewhat akin to QTC. And so when we tested the coolants themselves, we found that they minimally affected beat rate on their own, and they didn't really affect field potential duration on their own. Shown here is WS3 with beat rate on the x-axis and field potential duration on the y. But backing up right here, with co-treatment with norepinephrine, we found that actually addition of these coolants slowed beat rate and prolonged field potential duration. So it appeared to do so in a fairly typical manner that you would expect according to the beat rate to field potential duration relationship. But when we applied our field potential duration correction formula, we found that actually menthol significantly shortened and WS3 significantly shortened the field potential duration, indicating accelerated repolarization in these myocytes, independent of any change in heart rate. So those discordant observations suggest that the impacts of the e-cigarette aerosols, the coolants, are divergent when it comes to a whole organism versus the organ itself or its cells. But suffice it to say, our evidence indicates that coolants and nicotine salts exacerbate e-cigarette-induced sympathetic dominance and arrhythmias. The changes that we observe in heart rate and heart rate variability, as well as in repolarization, can predict e-cigarette-induced arrhythmias. And there's a multitude of other arrhythmogenic factors to consider. I didn't have time to elaborate on some of our kind of susceptibility models. We've been doing studies with my PhD student, Brittany Reynolds, on a model of pregnancy and exposure during pregnancy, and had some very interesting observations, including persistent arrhythmogenesis that unfortunately I just can't share with you today. And there are a number of remaining gaps, including the toxicity of countless flavorant chemicals and additives, as well as sort of the interactions with aldehydes, the role of reflexes versus direct toxicity to cardiomyocytes, and the translational relevance of all of this in the first place. Obviously, we're working with mice mostly, and it's not entirely clear if these observations can be recapitulated in humans. Moreover, long-term effects are not clear from our research yet, especially when compared to smoking. There are a lot of implications of our findings for both regulatory bodies and potentially for clinicians, in that e-cigarettes could acutely increase cardiac risks depending upon their ingredients, et cetera. And heart rate could be a risk biomarker for e-cigarette-induced arrhythmias. Long-term e-cigarette use can induce persistent electrical remodeling of the heart and/or autonomic dysregulation. And limits to e-liquid constituents could protect public health. So with that, I thank you very much for your time and attention, and open the floor to any questions.
Thanks so much, Dr. Carll.
We'll
open
Dr.
it up
Jones,
to,
if you want to start off.
yeah.
Did
So if you
have a
question,
any questions for to start off with Dr. Jones, or?
Yeah, I got it, Jason. I know how this stuff works, man.
Oh, yeah.
We'll be okay. Just relax. So yeah, if you have a question, just unmute yourself, or if you're in a position where you can't talk on the phone, please type. It just takes longer, Jason, for people to kind of click on and click off, and goodness, if they have to type something, it'll obviously take a while. So Alex, while they're collecting their thoughts, you mentioned at the very end some of the work that you guys are doing in pregnancy. I'm wondering, beyond otherwise healthy mice, right? Because there'll be some component of the user base of e-cigarettes that have other lifestyle factors. Such as diet and physical activity or inactivity. Any idea how those might come to bear? I'm thinking particularly in the realm of physical activity, which should kind of directly impact autonomic nervous system tone.
Yeah, great question. And that's something we're very much interested in pursuing in a subsequent proposal. The role of physical activity and potentially buffering against some of the autonomic impacts. But it's unclear. It may be that increased physical activity enhances sensitivity. I expect the former rather than the latter. But it's certainly of interest, especially because it's a practicable sort of, I guess, defense against self-harm if you happen to be addicted to e-cigarettes. In terms of diet, we have incorporated a couple of different diet studies.
What's that? It's pretty cold outside. Oh, yes, big time.
Oh, sorry.
Just keep going. That's Dr. Gopgazi accidentally unmuting himself. Just keep going.
That's all right. We have incorporated a couple of different experimental diets, including a high-fat, high-sucrose diet into this e-cigarette model, as well as a hypokalemic diet, one with low potassium. And we've seen, some encouraging results, but some of the problems with the high-fat, high-sucrose diet are that the telemetry signals can be very challenging to analyze. I think with the obesity and just the accumulation of fat, the signal gets kind of crummy. But we have seen limited evidence of a slightly enhanced arrhythmogenicity in that model. And then we've seen mixed evidence with the hypokalemic model. So moving forward, I'm planning on incorporating models of long QT syndrome, especially long QT syndrome 1 in the mouse, and long QT syndrome 3, which we're developing and phenotyping presently.